Behavioral Neuroscience, Chapter 2, Segment 1. Thoughts, feelings, behaviors, and almost everything that defines one as a person is determined by the brain. The entire body is in communication with the nervous system. Each neuron is a discrete information processing unit that communicates with other neurons. Glial cells are non-neural brain cells that provide structural, nutritional, and other types of support to the brain. Golgi believed neurons were endlessly connected like a tube, but Cajal proposed that a tiny gap between the neurons kept them separate. The neuron doctrine was then proposed. It stated that each brain cell is distinct structurally, metabolically, and functionally, and that information is transmitted across gaps between them called synapses, named by Sir Charles Sherrington. There is thought to be a quadrillion synapses in the human brain. Neurons contain mitochondria, which produces energy, cell nuclei contains genes encoded in DNA, and ribosomes, which translate genetic instructions from the cell nucleus into proteins. Extensions of the neurons, called dendrites, serve as an input zone, which receives information from other neurons across synapses. The cell body contains the nucleus and serves as an integration zone, analyzing the received information to determine whether or not to send a signal of its own. The axon serves as a conduction zone and carries the signal away from the cell body. It may split into branches called axon collaterals. Axon terminals are at the end of the axon and serve as an output zone by transmitting activity across the synapse to other cells. Multipolar neurons have many dendrites and a single axon. They are the most common type of neuron. Bipolar neurons have a single dendrite at one end and a single axon on the other. They are common in sensory systems such as vision. Unipolar neurons have a single extension or axon that branches in two different directions. One end is the input zone and the other is the output zone. They transmit touch information from the body into the spinal cord. Motor neurons govern movements. They have long axons reaching out to the synapse on muscles that contract in response to commands from the brain. Some contact and control organs and glands. Sensory neurons carry messages from the periphery back to the spinal cord and brain. Interneurons make up most of the brain. They receive information from other neurons, process it, and pass the integrated information to other neurons. Arborization is the elaborate branching of the dendrites of some neurons. Information is transmitted from the presynaptic neuron to the postsynaptic neuron. A synapse has three principal components: the presynaptic membrane of the axon terminal of the presynaptic neuron, the synaptic cleft, which is the gap that separates the two neurons, and the postsynaptic membrane on the surface of the dendrite or cell body of the postsynaptic neuron. Presynaptic axon terminals contain many tiny hollow spheres called synaptic vesicles. Each contains molecules of a specialized chemical substance called a neurotransmitter. This is what the neurons use to communicate with postsynaptic neurons. The neurotransmitter binds to postsynaptic receptors which react to it. Electrical change occurs in the postsynaptic cell. Dendritic spines stem the dendrites and allow for more synaptic connections. Now we move to box 2.1, visualizing the cells of the brain. Histology is the scientific study of the composition of tissue. Scientists use staining procedures to study neurons. Nissl stains are cell stains that reveal all cell bodies by staining RNA. Golgi stains are cell stains that fill a small proportion of neurons with dense dark product. They make the shapes more distinct. Autoradiography is a histological technique that shows the distribution of radioactive chemicals in tissues. Immunohistochemistry is a technique in which labeled antibodies are used to visualize the histological distribution of specific proteins. In situ hybridization uses radioactively labeled lengths of nucleic acid to label only those neurons in which a gene of interest has been turned on. And finally, a track tracer is a compound used to identify the roots and interconnections of neuronal projections. This could be anterograde or retrograde. The axon arises from the axon hillock, a cone-chain projection of the cell body. This is the axon's integration zone. Cone-shaped, I'm sorry. To innervate is to provide neural input. Important substances needed at the axon terminal are loaded into transport vesicles, which are guided through the axon by motor proteins. Axonal transport is the transportation of materials from the neuronal cell body to distant regions in the dendrites and axons, and from the axon terminals back to the cell body. The axon has two functions: rapid transmission of electric signals along the outside of the axon, and slower transportation of substances inside the axon to and from the axon terminals.